Hello and welcome to another episode of Momento Malum today. As of right now, the general consensus on the limiting factor in black raspberry cultivar yield is the limitation of genetic variation within these cultivars. In order to test this hypothesis, Dr. C.A. Weber of Cornell University used RAPD markers to detect the genetic diversity within the black raspberry cultivars selected for a study. The paper is called Genetic Diversity in Black Raspberry Detected by Rapid Markers. It was published in 2003 in Hort Science, and through this paper, he discovered the genealogy and family tree of the current black raspberry cultivars in play. This family tree will be shown in the last slide of this video. Let's begin. The black raspberry is a very niche group of species of blackberry, whose large-scale production is limited by the lack of superior cultivars for commercial production. Even with the demand increasing, the lack of suitable cultivars has continued to stymie and limit the growth of this industry. In addition, disease pressure has increased, and the most utilized cultivar, Munger, has been diminished in field lifespan. There is very little variation in harvest season, fruit size, or disease resistance, or other characteristics between individual cultivars. Various models on why this is happening have been created. The first is a lack of effort in breeding new varieties of black raspberry. The second is a lack of genetic diversity within the species itself, due to it being a predominantly self-pollinating species. Although the researcher did not desire to answer the question of why this happened the way it did, he decided to utilize the rapid marker genetic technology in order to analyze the amount of variation that exists within a sample of the cultivars utilized by North American growers. The researcher utilized 16 black raspberry genetic lines, 14 commercially grown cultivars, and 2 wild specimens, as well as the addition of 1 blackberry hybrid and 2 raspberry cultivars. As a method of comparison, the genetic analysis was done through the Phylib Suite phylogeny software package, using the REST-DIST program and the Neighbor program. It's through this program that the genetic data was analyzed and the results were sent out. And here are the results. Hello, I just want to let you know, if you want to fund this channel, you can do so on the platforms seen in the description below. The hypothesis that the genetic diversity of the black raspberry being inadequate for further improvement, both in the commercial and wild populations, has been confirmed. As a whole, the wild and domesticated genotypes are 81% similar to each other on a genetic level. Comparatively speaking, within domesticated red raspberries, the cultivars are 70% genetically similar. And then in a study by Dr. Oraguzzi, it was found that a sample of 36 cultivars had a range of 52% to 70% genetic similarity between one another. So yes, on the whole, the gene pool of the black raspberry is very, very limited, especially compared to other wild species and domesticated fruit. 58% of the variability, however, has been found in 5 genotypes of black raspberry. These are Black Hawk, Cumberland, John Robertson, NY9960, and NY9961. Black Hawk is a cultivar derived from 3 of the 4 genetic lines of wild black raspberry utilized to make most of our varieties. Cumberland was selected from a wild black raspberry, as was John Robertson, and both NY selections were obtained from the wild themselves. This indicates although the genetic variability within the species is low, it is somewhat higher within the wild species. Although this seems to be not always the case, as several wild selections such as Early Sweet, Hanover, and New Logan seem to have the same level of genetic variability as a regular domesticated version. And none of the 16 cultivars were more than 2 generations removed from the wild ancestors. The researcher concludes that it's plausible that many of these so-called were just escapees from gardens and were not truly wild specimens but rather feral specimens. The 11 least variable black raspberry cultivars were collectively 92% genetically identical, which means only 8% of their genes, which is quite a tight gene pool. The genetic distance between a red raspberry cultivar and a common black raspberry is 40%. In 1996, Dr. Daubeny suggested that the genetic diversity in black raspberry might be found by sampling genotypes from the extreme ends of different populations within its natural range. The variability found by Dr. Nybaum and Dr. Schall in 1990 seems to confirm this hypothesis. Crossbreeding between species to incorporate new traits may also be a valid way of improving the gene pool. The two NY cultivars, both selected from the wild populations, have semi-smooth canes and yellowish fruit, not typical for the black raspberry. And Cumberland is said to be partially blackberry in its pedigree, due to the leaf shape being intermediary between a blackberry and a black raspberry. This may indicate that the addition of hybridization between other species of rubus may incorporate new traits and expand the gene pool, although it does not always work that way, as the early sweet cultivar is not very variable, nor is Black Knight, and both have minor traces of another species called Rubus leucodermis. Cultivars edge of the range of the black raspberry, such as Kansas and North Dakota, show great variability, as John Robertson and Black Hawk were both selected from specimens from those regions. The researcher concludes by saying, it's possible that we could increase the genetic variation by sampling the wild specimens from a broader range of the species population across North America. Combined with integressions from other species may increase disease resistance and fruit quality, as well as yield, and may lead to the creation of newer and better cultivars. And that about covers everything. Thank you for watching.